Manjunath sir, shall we start?
Okay, madam.
Sir, can you switch on your video for some time?
1 second.
Good morning to all the participants. Welcome to today's session of FDP. Our speaker for today is Dr. Manjunath Reddy. He is Associate Professor in the Department of Biotechnology, R.V.College of Engineering. Sir will be the speaker for this session. He will be talking about SAS and related programming aspects in biostatistics. Sir, on behalf of the staff, students and management at the Biotechnology Department, the Oxford College of Engineering, I take it a privilege to invite you and welcome you to this session, sir.
Thank you, madam.
And sir, you may please start with the session.
Okay, madam. Thank you, madam. Thanks for the introduction. Good morning, everyone. I know everyone is mute here. No worries on that. You please listen carefully. If you have any questions, you can put it in the chat box, and also you can ask me later. Okay. So we will be talking about the bioanalyticals, or the analysis in biology or biological data, we can say. So my title goes like this. This is the analytical tools and techniques for biological data, and also I will be emphasizing on SAS. Okay, so probably you might have heard what is an SAS and all. Probably.
Excuse me, sir.
Yeah. Yes, madam.
Could you put your presentation on slide show mode, sir?
Yeah, I am doing it, madam. I have shared now.
No, sir. We are only seeing the PPT mode.
Okay. One second. It is in presentation mode only, madam.
It is in presentation.
Yeah.
No, sir. We can see your slides on the other side also.
Okay.
Yeah. If you could unshare and then share again.
Yeah. Doing it. I think now it is clear.
Yes, sir, it is clear. You may proceed.
Okay. So initially, I'll be talking about this biostatistics, how exactly what is important, and probably but I will learn some of these concepts, but still I would like to revamp certain things. And after that, I will be emphasizing on SAS. Okay, for biological things or biological data, how it is different, why we have to give more emphasis for the biological data, whether it is really required or not, why any analytical tool can help why not any another analytical tool can does this analysis or not, how best we can interpret, and all such stuff that we will discuss today. We have time limit for 2 hours today, so let us make use of these 2 hours efficiently. Let me come to one simple thing. We know data is everywhere, data is piling up every day, and for today there is a kind of some type of data will be there, and tomorrow it will be enhanced, it will be more. So we need some more and more analytical tools so that we can do some interpretation. Before going to that, another aspects, I would like to define what is the statistics or biostatistics. Listen carefully some of these terms. Biostatistics that you can define something like it is the collection, organization, analysis, and interpretation of the data. Okay, so the terms are something like this: collection, organization, analysis, and interpretation of the data. So collection of the data will happen in different way.
Excuse me,
When,
sir.
yes, madam.
Your slides are not changing, sir.
No, no, madam. I'll go to the next slide
Yours is
afterwards.
still in the first slide only.
Yes, 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 madam. I have not
Okay.
put it, madam.
Right, sir. Okay.
So I just wanted to give the definition in the beginning, and then I'll move on to the other things. So I was telling about the collection, organization, analysis, and interpretation. So in all these aspects, the biological data vary. So collection, the sampling, either the sampling or the population methods are going to vary. And the interpretation at the end is going to differ from other type of the data. Okay, moving on to the next slide. And these are the things that I would like to discuss. Maybe superficially we will discuss on these topics, and finally we will discuss more on the SCAS. Okay. My topic is something like not in detail. I'm going to talk on this. It's on data, measures of central tendency and the spread, graphical methods, sampling methods, variance, errors, range, bias, and such kind of things. Hypothesis testing. So it's nothing but significance. What is an hypothesis and how best the hypothesis can be tested, and what are the probability values? How do these probability values can be calculated or interpreted? Okay, student test, student t-test, chi-square test, z-test, and ANOVA. Probably you might have heard about this, but even you may even know how to calculate this also. But I will not go in detail about how to calculate, but I will try to tell you about how where all these can be used and how the interpretation can be done. Another set of information for the discussion is the probability and the distribution. Probability and the distribution. Oh, and what is probability, loss of probability, normal, binomial, and are various types of the probabilities that we can discuss. And if you want to see relationship among the parameters, which are the things that we can discuss. So it's about correlation coefficient and regression analysis. Okay, the four red colored things that I have put it over here. The first thing about the introduction, that will talk about the data, various types, whether continuous, discrete, and such kind of the things. Next is the hypothesis testing. Next, the probability and distribution of the probability, or the distribution of the organisms, or the distribution of the things are present in this study or the research or something of that sort, and how to find the relationship between the organisms or the things or the data that we can discuss now. Okay, statistics definition I had told you. Biostatistics is nothing but the application of these statistical techniques or the tools or the methods to the biological data. Why it is different? Why not? Why we have to call it as biostatistics? Why not just simply as statistics? Of course, statistics have got some numericals. Statistics have got some kind of the data, and that data is different from one type to another type. But why we have to concentrate more on bio is 1 plus 1 is not equal to 2 in biological setup or biological system. Okay, how it is possible? Probably you might have seen in your laboratory and all how the micropropagation will take place, how the cell divisions will take place, what are the types of the cell divisions that will be there. So, and maybe the population explosions from one level to various levels. You can see biological data is different from other type of the data. Okay, there is no linearity in biological data. In all other cases, you can find some or the other way the linearity in the data, but here in biological data the linearity is very minimal. Once this is minimal, probably you cannot give the final conclusion or the decision or the interpretation like you are going to give it to some other disciplines. That's why the biostatistics is very, very important. Coming to the data is nothing but the arrangement of the numbers, arrangement of the information, or you can call it a set of information. Okay, data is a plural term, guys. Datum is the singular term. Okay, so for that, different types of the data are there. It is for the convenience of the study, convenience of the experimenter, okay, or the instructor. It is like continuous data or discrete data. You can categorize like categorical data and numerical data. You can even categorize like nominal data as well as ordinal data. Continuous is continuously present. Discrete is it is discontinuous or it is not continuous. Okay, very simple. 1, 2, 3, 4, 5. 1, 5, 10, 15, 20, 25, 30. 100, 200, 300, 50, 100, 150, 200. So these are have got some regular range, regular type of the continuity. Okay, they are called as continuous data. Discrete data may be 1, 10, 11, 15, 12, 13, 25, 100, 150, something of that sort. The data will be there. And its arrangement is also discrete, it's not continuously present. You can call it as some of the data as categorical data and numerical data. Probably in any kind of the information, what is the height of the individual that you can ask? How many number of the people are up to the height of 5 foot that you can ask? Okay, but if you define some of the characteristics, maybe the colors, okay, so there you will find some categorical data. Maybe if you say the different types of the people, or maybe different types of the plants, or maybe different types of the animals, okay, so you are going to categorize the lions or the tigers or the deer, something of that sort. So they are the categorical data. Generally you might have seen in your data set f of x and all. Okay, there you can apply this categorical data applications. And numerical, all are numerical only. Many of the times you may not get the numericals, but in that case the numericals can be converted to the non-numericals can be converted to numerical and analysis. Okay, so that is also one of the thing. Nominal and ordinal, this is another type of the classification of the data. Nominal is the names, okay, ordinal is the any kind of the numerical data. Okay, nominal is mostly the names are the heights or hillocks, something of that sort. Okay, so how do we get the data? How do we get the data? There are two major types that we can follow. One is the sampling method, another one is the population estimation. Okay, these are very simple things. Probably you would have heard or would have learned in your previous standards also. But still, we will brush towards the biostatistical applications way. Okay, sampling. How do we? What is sampling? It is a subset of the population. Okay, what is called as population? The population is not just only the human population, guys. It is the population of animals, or the plants, or the microorganisms. Okay, of various sectors, or of various kingdoms, or various phylums, or so on. Okay, so the sampling is nothing but a subset of the population. So you cannot estimate the entire population. So what you do, you are going to estimate a subset or a sample or a few of the population that's going to be called as sampling. Okay, there are various types. Okay, let's not get into much about that, but there are various types of the sampling methods. Why? It is for the convenience of the study, first thing. Another thing is to reduce the bias. Okay, so we will have some of the logics. We can say based on this logic or based on this existence, based on our previous knowledge, based on our library information, we can define or we can design some of the sampling techniques. Okay, so it may be cluster sampling, it may be random sampling, it may be non-random sampling. Okay, it may be a convenient sampling, it may be just a quadrant sampling. So like that, there are various types of the sampling techniques are there. Okay, sample and population. Sample is subset, as we know. Okay, population is the entire population that you are going to consider. For example, there are 60 students in the class. If I want to know whether my lecture is good or bad, I just don't need to go to everybody and ask whether mine is good or bad, something of that sort. Okay, what I will do, I will just take sample. I'll just say, I will just ask a few people, maybe if you two students in the front bench, two students in the back bench, and two students in the middle benches. Okay, like that, I will take the sample and I will get some interpretation out of it. But if I want to distribute the chocolates to the entire 60 students, what I cannot give the sample like that. Two in the first bench, two in the middle, and two in the last bench. If I could give the chocolates to these people, the people may not understand. People may not give the correct interpretation. So then what I have to do? I have to go for population technique. Population is nothing but the entire population, the entire all the individuals will be considered in that set or in that data. Okay, population definition is something different. Since you're all biotechnology students, so I wanted to define tell you about what is the population definition. Population is nothing but number of individuals belonging to one species at a particular unit space and area, unit at a particular time also, and they are capable of producing the young ones at their adulthood. Okay, they are called as the population. In generally, for statistical purpose, when you talk about the population, you have to consider all the individuals. You should not leave anybody. Okay, you should not omit anybody. You should not say, of course, we can leave this person, we cannot leave this person. Not like that. You have to consider every individual in that. Okay, so it's nothing but the population. So below that I have written DF. DF is nothing but the degrees of freedom. Degrees of freedom is nothing is having some different meaning. That's why I have written degrees of freedom is nothing but the freedom of the individuals that are present in one set. At least those number of the individuals are present. Okay, it will be something like this. X in the below the X we will write 10 values. Say suppose 1 2 3 4 5 6 7 8 9 10. This is one set of sample. Okay, this is one array or one set of data, right? So in this case, the degrees of freedom is equal to n minus 1. It means n is equal to 10 and 1 is just because it is one set, we are subtracting 1 over there. So that is n minus 1. It means that at least 9 values are freely accommodated in that data set. Okay, that is the degrees of freedom. But that is only applicable only for the sample. Okay, because in the sample there may be kind of bias, there may be kind of error, because if 30 students are doing some sampling estimations across Bangalore, not essentially everybody should get every kind of the information at 100% accuracy. That may not happen. That may happen or may not happen. Each individual perspective is different. Each individual subset is different. Okay, for that matter, there may be kind of errors that will be existing. That's why there we have to understand the degrees of freedom. So in say suppose x have got n is equal to 10, it is n minus 1, that is nothing but 9. At least 9 values are freely located in that data set. Okay, but for the population, you should not consider the degrees of freedom. There is no degrees of freedom for the population. It is applicable only for the sampling. So the sampling may be of two different types, like random and non-random methods. Random may be simple sampling. Random sampling is also called as probability sampling. It may be the simple sampling, stratified sampling, convenience sampling, or the quota sampling. Based on different perspective, based on the requirement of the study, we can take or we can consider or we can follow these kind of the sampling techniques. Non-random is is the one where you can have to consider the all individuals in the population. Then it's called as non-random. It is a specified kind of the sampling technique. Coming to the representation of the data, it's very easy, and you have done this in your school days also. Okay, it's very simple also. Tables and figures representation are sometimes in the in the form of text also. Okay, data is not essentially the numericals only, but it is more of numericals, right? So there may be sometimes there is a text messages also. They are nothing but the data. So generally, or more often, we represent the data in the form of tables and figures. Okay, and figures may be the graphicals may be the line graph, bar chart, you know, um, like pie charts, okay, heat maps, so on, so forth. Okay, so there are various types of the figures or the graphical representations that you can see. Kurtosis and skevedness. Okay, so just imagine there is one bell-shaped curve. Okay, just imagine there is one bell-shaped curve. So there is this bell-shaped curve. If you cut it into two pieces in the vertical way, so it should come in the middle that you have to cut. Okay, so that is the normal distribution, right? That is the normal distribution. If that peak is moving towards the right or maybe moving towards the left, then what happens? It is called the kurtosis. Okay, maybe left is a leptokurtic. Sorry, um, you know, I'll come back. It's not the kurtosis, it's the skevedness. Okay, left skewed and right skewed. If it is towards the left, then what happens? The x values are in the beginning itself. It has got highest peak, and later on it has tapered. If it is tapered in the beginning and highest in the right, then it is right skewed. Okay, so kurtosis. Suppose if you push your bell-shaped curve down, okay, so it may be a flat kind of the curve that you can see. Then it is called as platykurtic. Little up, mesokurtic. As you can imagine, the bell-shaped curve, that is the leptokurtic uh, curves. Okay, so that shows the distribution of the individuals. That shows the distribution of the individuals in a population or maybe a sample. Okay, so it is like that. So the representation of the tables and figures are very important in the data analysis or to find the interpretation out of it. How? It is not just to make sure that they are good enough. Okay, it is also to say that large data can be put into a smaller image kind of the thing, so that you can save some space. It can be attractively seen, visible also. Okay, so measures of central tendency. So these things also you might have learned. So you might have exercised it in your biostatistics class. You probably might have done some experiments or calculations on these things: the mean, median, and mode. I told you, if you cut the normal shaped curve, normal pop distribution curve, okay, the bell-shaped curve, if it cut exactly into two halves, in that case, mean, median, and mode are going to be placed in single line. Okay, it is like that. The mean is also called as arithmetic mean, or just a mean, average. Okay, it's also called as an average. There are various types. The mean is mean, or arithmetic mean, or the average is something like it is the sum of the values in a data set and divided by n. It's very simple. You just sum up all the values and divided by n. Say suppose there are 5 values, 1 2 3 4 5. Okay, you add all the values, it will become 25. So then divided by 5 is nothing but sorry, it's not 25. 1 2 3 4 5 15. divided by 5 is nothing but 3. Okay, so 3 is the mean value, right? So like that, mean, median, and mode are can be calculated. Median is the central value. Okay, you know, when you will ride a bike or something of that sort, so the road will be divided by the road divider, and that's nothing but a median. Okay, so that is the median. So centrally located values. So there is a data is present everywhere. What will happen? You are going to um, put all the values either in ascending or descending order, and centrally located values that you are going to take. Say suppose the data is 1, 2, 3, 4, 5. The centrally located value is the 3. Okay, so it may be, say suppose 1, 2, 3, 10, but still 1, 2, 3, 4, 10, but still centrally located value is 3. Sometimes if there are 4 values, say 1, 2, 3, 4, then what happens? Centrally there are 2 values, 2 and 3. There are 2 values. What you will do? So 2 plus 3 you will add and divide by 2. 2.5 is the centrally located value. That's the median. Okay, like that. The mode can be calculated like if there are any value which is repeated. It's as simple like that. In a data set 1, 2, 3, 4, 4, the 4 is the mode. Okay, so because it has more weightage in that data set. Okay, it has more influence in that data set, and that's why it is the mode. Measures of central tendency, that is the heading that I have given. Measures of central tendency, it means that the centrally located value that you are going to measure. Okay, so that value have the capacity to tell, or the capacity to represent the entire value, entire data set. Okay, they are the measures of central tendency. How that is deviated, whether it has too much deviation or less deviation. Okay, for that there is some technique, that is the measures of dispersion. Okay, measures of dispersion. Here you can go with the variance, deviation, and standard deviation. Okay, variance. So there is a mean. You know how to calculate the mean. With that, how the first value, second value, third value, fourth value, fifth value, like that, n number of values are going to be varied. That is the variance. Deviated. That's the deviation. So when you calculate all the variance or the deviation, so there may be chance of plus and minus values, occurrence of this plus and minus values, and there will be altogether it may be a zero value, or maybe slighter minus or positive values may be obtained. For that, what we will do, there may be chances of nullifying there should be nullifying of the negative values. What we have to do for the nullifying the negative values, if there are any minus values in any of the data set, what we have to do, we have to square it up and take the square root of it. So that's the simplest way of nullifying the minus values. So that can be possible by conducting the, or by using the standard deviation. So the standard deviation formula itself is nothing, is like that. Okay, the whole square root, in that whole square root, in the numerator, sum of x minus x prime whole square, and divided by n minus 1 for the sample and n for the population. Okay, it is like that. There are two more aspects, like the range. Say for example 5 to 10, 10 to 15, 15 to 20, like that the range will be there. Lower limit, in that case lower limit is 5, upper limit is 10. Next one, 10 to 15, 10 is the lower limit and 15 is the upper limit. 15 to 20, 15 is the lower limit and 20 is the upper limit. It is like that. Okay, so range over there is 5. Okay, 5 is the value. Say suppose 5 to 20, 5 to 10, 10 to 15, 15 to 20. See, this 10 is going to be repeated. Oh, it is there in the as a lower limit. It is there in the upper limit also. 15 is there in the lower limit. 15 is there in the upper limit also. In that case, what you have to do, you have to consider one way, one single way. Either you have to put it in a uppercase or in the lower case. To write, you can write like that, but for a consideration of its values, either you have to consider in the upper limit, consider as the upper limit, or in the lower limit only. In one case you can consider as the upper limit, and on another case you can consider as the lower limit. That is not possible. Okay, that is not the correct way of representing the range. Okay, whatever we do, there are various types of the errors. Okay, various types of the errors that you can see. What are the different types of the errors? What do you mean by an error? So an error is something like, that is an unnatural way of presentation. That is an unnaturally present some of the data that is present in the data set. Okay, it is not required. It may be inevitable, but it is not. Its influence is not required. Its occurrence might be there, but its influence is not required. We have to ignore. We have to rectify it, or we have to nullify it. There are, because of these errors, the data is not called as properly generated, or it's not proper only. There are different types of the errors, type 1 and type 2 errors, that we will discuss later also. But the errors can occur in various levels. In sampling methods, there must be some errors. In data collection points, there may be some errors. In human perception, there might be some kind of errors. In calculation ways, there may be some kind of errors. In data entry, there may be some kind of errors. In experimental ways, if you consider the experimental data in the chemistry lab also, there may be errors with respect to the titration. There may be errors with respect to the chemicals. There may be errors with respect to the power, that is power supply, and lot more. Okay, so because of all this, the data accurate data cannot be obtained. So what we have to do, we have to reduce the error. How to reduce the error? For example, SE is the designation for standard error. SE. Okay, guys. I'll just tell you the formula of it. Then it will be easy to how to reduce the error and all that can be easily understandable. Okay, SE is equal to x prime divided by square root of n. Okay, this is the formula, right? So x prime is the mean or average or arithmetic mean of the values, and square root of n is the number of samples or number of trials or number of experiments or the repetitions that you have done. Okay, to reduce the error, SC, it should be reduced. SC should be very, very minimal. Okay, in that case, what you have to do, you have to increase the number of samples. If you increase the denominator, automatically the SC is going to be reduced. Okay. Coming to the probability. The probability is something like, probably today I was expecting 50 members to attend this class. Probably I can reach the class by 9 o'clock. Probably I can skip the Silkport junction, or probably I will get signal very fast in the Silkport junction. So something like this. There are various ways or various places we will use the term probably, right? So it means that this probability, the occurrence of that data or the occurrence of that particular data in that data set, how it is occurring, whether it is yes or no. Very, very simple kind of the examples in a probability distribution might have been, you might have observed, like tossing of a coin. Either you will get a king, head or a tail. Okay, and in gambling, it's more of usage of the probability that can be observed. Maybe king, queen, joker, and various numbers, various types of the designs. So all these things are going to give, multiple ways of understanding of the probability. If you roll a die, so what is the probability of getting 1? What is the probability of getting 2? Because there are 6 phases.
Okay, there are two different rules. Okay, one is additional rule, another one is multiplication rule. Okay, in some places we are going to add the probability values, and in some cases we are going to multiply the probability values. Okay, these probability values are in short, it is also called as p values. Okay, so these p values may also be obtained in the Reddy reckoners or the Reddy references. Okay, so some of these values have been calculated, and these are called as the Reddy reckoners or the Reddy reference values. Okay, so these values can be considered, or these values can be compared with our experimental values, and we can give some conclusion or the interpretation. Okay, it is like that. And in the probability, in a major way, there are three types of the probabilities. Okay, as there are three types. One is the binomial probability or binomial distribution. Another one is the Poisson distribution or the Poisson probability. The third one is a normal probability. There are various other ways also. There are various other different types of the probabilities also. Okay, but in a major cases or in majority, people will be using these three different types of the probability distributions. Okay, binomial. Okay, so it's nothing but two. Say tossing of a coin, it has two faces, so you'll expect either head or tail. Say you are attending the class or not, yes or no. Okay, are you pass or fail, pass or fail, so two events. Okay, like that, there are two ways of representation. Okay, so then it is called as binomial probability or binomial distribution. It may be binomial distribution, but it is continuous. It may not be binomial distribution, but it is continuous. For example, one simple thing I will tell you. In a silk board junction, how many number of vehicles will be crossing per second or per hour or per minute? If you could calculate, so there are four different approaches or four different directions the vehicles are going to be approached towards silk board signal. So how many number of the vehicles in this left side, right side, opposite side, north side, east side? Okay, if you could calculate per minute or per second or per hour, something of that sort. So that probability distribution is nothing but the Poisson probability. Okay, so that's one thing. Another one is the normal distribution. Normal distribution. It is very, very normally located distribution. Okay, so how on that? Simple. So wherever you are sitting and attending this class now, just think of there is one statement, okay, or there is one value that is the oxygen that is present in the environment is 21%. Right? Do you agree? It is an average value. Wherever you are sitting in that room, no, just imagine oxygen is present everywhere. That is the 21%. Okay, is it true? If that is present everywhere, it's 21%. That is a normal distribution. If it will not obey that, so then what? So okay, so for that there is a one more technique or the tool. There is nothing but the hypothesis testing. There are two hypothesis testing, or the two hypotheses that we can make. One is null hypothesis, another one is alternate hypothesis. The null hypothesis, null means zero, no hypothesis. No, it's not like that. It's not no hypothesis. It is there is no distribution, or there is no difference in the distribution, or it is going to obey the normal distribution. Okay, so I'll take the same example. If the oxygen, that is whatever the calculation of the oxygen that you are going to make in your room, if everywhere in all the nook and corners of that room, everywhere if it is only 21% that is present, then it is normally distributed. And your hypothesis also holds good. The null hypothesis holds good. There are no differences. For example, it is not like that. If you get the data 18% in one place, 21% in another place, 22% in another place, sorry, maybe 10% in another place. If that is the kind of the distribution of the oxygen concentration in your room, then it is an alternative hypothesis. Your null hypothesis is not holds good. It is not normally distributed. There are various differences among the values. Okay, so it is like that. And while discussing the errors, I was also talking about the type 1 and type 2 errors. Okay, so here is one simple table that is here: actual situation truth, okay, and decision how to make the decision. Okay, H0 is the null hypothesis, and like this, H1 or HA is the alternate hypothesis. If do not reject and it is true, okay, H0 is true, then 1 minus alpha, and correct, it is the correct decision. Okay, if it is incorrect, it is false, H0 is false. Okay, but you are going to reject the H0. Okay, so then incorrect decision. See, H0 is true, but still you are going to reject it, then it is incorrect decision. Type 1 error is going to occur. If it is false and then reject, then it is type 2 error, and you are going to represent with the beta. Type 1 error, you are going to represent with the alpha. At the end of the class, I will let you know what is alpha and how to read about alpha and beta. If H0 is false and you are going to reject H0, if the correct decision is made, then you have to consider it is 1 minus beta. Okay, alpha is type 1 error and beta probability is type 2 errors. Okay, its values are nothing but 1 minus whatever, either alpha or beta. Probability values, either you will sum up or addition or multiplication, altogether the final value must be less than 1 or less than 1 only. If it goes beyond 1 or more than 1, you can always say, or you can always tell that that person have done something wrong in that data calculation. Okay, there are different types or steps in the hypothesis testing. This is not visible properly. Okay, so in this hypothesis testing, first you have to make the alternative hypothesis. Okay, state and mention the alternate hypothesis. Levels of significance that you have to mention, and test the differences. Okay, and rejection range also. Once again, the letters are not visible. Can you give me 1 minute time so that I will make it right and come back? In front of you only, I am going to change it.
So, is there a technical problem, sir?
No, it's not technical problem. The font size is too big, so I just need 30 more seconds, madam.
Okay, sir.
Yeah, it is done now. I'll just share the screen. Yeah, steps in hypothesis testing that we are discussing. Okay, so these are the 7 different steps that one can follow, even if you can omit 1 or 2 steps, but all these things that you have to follow. First thing is state the null and alternate hypothesis. You have to mention what is your null hypothesis and what is your alternate hypothesis. Next, you have to say what is the level of significance. Okay, so what is the level? Either at 0.5% you are going to estimate, or at 10% you are going to say, or 5% you are going to estimate. Like that, you have to mention well in advance. Okay, so it is like that. Then determine the test distribution. So what is the distribution? Which test you are going to do, whether it's an ANOVA or Z test, or which type of the test that you are going to do. Okay, so that you have to mention. Define the rejection and critical range. At what range you are going to reject, at what range you are going to accept the null hypothesis, that you have to mention well in advance. Okay. Next comes the state the decision. You have to make the decision and state it, mention it. Then make the necessary computation. Here comes what kind of the computation or the analysis that you have to do. Okay, either it may be a Z test, or it may be T test, or a chi-square test, or ANOVA. In ANOVA there are various types. Okay, so which one you have to do that computation, you have to do it, and then make the statistical decision or the interpretation, you have to do it. Okay, these are the different types of the hypothesis testing that you can follow. Okay, I told about these tests, no? So one is the Z test. Z test, very simple. X minus mu. Mu is the and divided by rho. Okay, mu is the mean of the population. X prime is the mean of the sample. Okay, standard deviation of the population. Rho is the standard deviation of the population. Okay, SD is the capital S is the standard deviation of the sample. Okay, this is a very simple, but a very simple formula and all. But when we have to do it, when we have to consider this Z test, okay, the sample Z size when it is greater than 30, if n is equal to greater than 30, then we can use Z test. Okay, and some conditions are something else like this. The data points should be independent from each other. The data points should not be dependent on. Okay, so and in other words, the one data point is not related to another, or it's not going to affect, it's not going to have any kind of the influence on the another data point. Then only you can call it, you can go with the Z test. Okay, so suppose 1, 2, 3, 4, 5, these values are there, but 1 is independent value, 2 is independent value, 3 is independent value. There is no dependency over there. There is no influence of the one value with the another value. So in that case, you can use the Z test. And it must be normally distributed. That's how we will start. Okay, that's how we will start the analyzing the hypothesis, but it must be normally distributed. If it is not normally distributed, whatever the alternate hypothesis that we have made earlier, that will be considered. Okay, so it may be anything. Alternate hypothesis may be anything. However, for larger sample sizes, it's like more than 30. This does not always matter. Then you can go. Okay, sometimes it's not matter because it's always in the normally distributed mode. Data should be randomly selected from a population where each item has an equal chance of being selected. Every individual in a data set must be in a capability of selecting itself. Okay, but you are selecting only a randomly a few subset or few kind of the individuals, and that's nothing but a sample. Sample size should be equal if at all possible. If possible, say suppose x is equal to x, the values are 5, y values are 5, z values are 5. Okay, then it is equally distributed values. Okay, within that value it may be different. Say suppose 1, 2, 3, 4, 5 is the x values, 6, 7, 8, 9, 10 is the y values, and 11, 12, 13, 14, 15 is the z values. Okay, but n is always same in all the three cases. Okay, so then we can go with the Z test. T-test formula is not very great. It's not very different from the result test. It's also simple and equal, almost similar to that of the result test. Okay, but what happens in the T-test? The T-test, the values must be less than 30. Okay, there are three main types of the T-test that are the features that you can think of it. One is an independent sample T-test compares the means of the two groups. Two groups may be different also. 1, 2, 3, 4, 5. Another one is 6, 7, 8, 9, 10, 11, 12. Okay, so there also you can use. If it is a paired T-test, okay, it compares a means from the same group at different times. Okay, it may be from this year, next year, it may be from IPL season 19, IPL season 20, IPL season 21, something of that sort. Okay, you're going to compare a one sample T-test. Test the means of a single group against the known mean. So suppose if you have any values which are already present in the library, so then what you can do, you are going to compare the your values with the library values. Like you are taking the mu value, mu is written over here, that is the standard values that you are going to consider, and the x prime is the experimental value, s is the standard deviation, and n is the number of values. S divided by square root of n is nothing but the standard error. Another type of the test, hypothesis test, is the chi-square test. Okay, the chi-square test is used to test a distribution observed in the field against the another distribution determined by the null hypothesis. Okay, there are two distributions, or two values, or two set of things, are the values, are the numbers are there. Okay, one is the observed values that are present in the field or the experiment or in the conduction of the process and so on, or maybe the calculation also. Another distribution is determined. It is already determined by the null hypothesis method. Okay, null hypothesis has been applied, and that value have been determined, and that is present either maybe in the library, or maybe in that system, or maybe that is in a particular process. Okay, so if you are going to test your observed values with respect to that of the your null hypothesis values, okay, then is there you can use the chi-square test. This very simple formula. Okay, this is the chi-square, the capital X square, that is equal to sigma of sigma. You just see it is for the entire set. Okay, O minus C O square divided by E. O is the frequency of observed values, and E is the frequency of expected value, observed as well as expected values. Okay, and interpreting the results. If above the critical value, then accept the hypothesis. Okay, if below the critical value, then reject the hypothesis. Hypothesis is nothing but the null hypothesis only. If it is specified or not specified, you have to consider only the null hypothesis. If it is specified only for the alternate hypothesis, then only you can consider the alternate hypothesis. Okay, that's why I have written like this. If above the critical value, suppose you are going to calculate one chi-square value. Okay, that value is 3.02. For your what you are going to do, you will compare this value with the table values that are already present. So which is the table value in every statistics book? So you have the table values. Okay, it may be t-test values. These values are nothing but the probability values. If you have any statistics book, you just go and see what is the table value. You have to see the backside or the at the last pages of that book, wherein in the annexures in all the statistics books it has been mentioned what is the t value, what is the alpha value, t alpha 0.05, t alpha 0.005, t alpha 0.01, t alpha 0.1 and 0.5, like that it has been mentioned. Okay, alpha is the type 1 error that we know, and 0.05, 0.005, they are all going to represent the error rates. How does the error is going to be calculated and all. If it is more accurate, okay, error is very, very less, then P alpha will be 0.001. Okay, this 0.001 into multiply with the 100. Okay, if you come to the calculate the percentage and all, it is nothing but 0.01. Okay, the 0.1 suppose. Okay, so from 100, if you could minus 0.1, it is nothing but 99.9. Okay, this 99.9 is the accurate value. The error rate is 0.1. Okay, I think hope you understood this. Okay, that's what I the beginning also I told, at the end I'm going to talk about this, right? So the probability value, which is already present in the table or in the books, okay, or in the libraries, that we are going to compare with the experimental value. If it is more, the experimental value is more than the table value or the critical value that is mentioned in the reference books. Okay, then you have to accept the null hypothesis. If it is less than, then you have to reject. Okay, probably about significance and insignificant. Okay, the significant is nothing but it is more than the value. Insignificant is nothing but less than the table value. It is as simple like that. Apart from that, there is one more kind of the analysis, that is the analysis of variance, the ANOVA. Okay, so ANOVA is nothing but analysis of variance, how it is varied from one population to another population. Okay, like that. Say suppose there are two populations, X and Y. Okay, population, two data sets are there, X and Y. Okay, so here there are differences in X itself. There are differences. Okay. Sorry. Say suppose you have the values 1, 2, 3, 4, 5. So there is a difference between 1 and 2, 2 and 3, 3 and 4, 4 and 5. Okay, and there is a difference between 5 and 3, 5, 2, 5, 3, 5, 4, like that. There is a difference. And another value is another data set is the Y. So in that Y, 11, 12, 13, 14, 15 values are there. Okay, in that itself, there is a difference among the values. And there is a difference between X and Y values also. The differences, okay, there is a variations or the deviations of the values that you are going to get in both the cases, within and between the sets, within sets as well as between the sets. Okay, the testing groups to see if there is a difference between them. One way has one independent variable with two levels, one way and one the healthy wala adu. Okay, for example, brand of cereals. Okay, two ways has two independent variables. One way have got one independent variable with two levels. Okay, two way have got two independent variables. It can have multiple levels. Okay, for example, brand of cereals and calories. If we talk about the brand of cereals, okay, so cereals, then we will just talk about the brand of cereals. You're not going to talk about the quality, quantity, and such stuff. If you consider the brand of cereals as well as the calories in that, okay, then it has got two independent variables, but it may have any number of levels, multiple levels, 2 levels, 3 levels, more, many levels. Okay, then it is nothing but the two-way ANOVA. Okay, it has to be written like this: one way ANOVA and two way ANOVA, not like this. That's why I wrote like I am representing like this, right? One way I have striken off here, two way I have striken off here. There is no mentioning of the number here. You have to mention this only. That is the rule. Okay, it depends on the number of independent variables. So you can define either one way or two way ANOVA that you are going to consider. This ANOVA formula is very, very simple, but to get that those values, you may have to do some more computations over there. This ANOVA is nothing but differences between the variables divided by differences within the variables. Okay, between is suppose X and Y you have, so the variations between X and Y variables you are going to calculate, that will lies in the numerator. But if you have the differences between X, okay, X1, X2, X3, X4, okay, Y1, Y2, Y3, Y4, so excuse me, if those kind of variations are there, so that will come in the within series. So that will be in the denominator. Okay, the V or the ANOVA is equal to variance between that between is above or in the numerator, and variance is within is in the denominator or below. Okay, so that is nothing but the ANOVA over there. What is that variance within and variance between? How to calculate that? So some kind of the formula are there. So it's I'm representing that formula over here. Sum of squares. B is for the SSB is for sum of squares for the between. SSW is for sum of squares for the within. Okay, so degrees of freedom, I told you in the beginning, degrees of freedom, how and where we are going to use. So since the standard deviation, from the standard deviation to any other calculations in the biostatistics, we will use the degrees of freedom. Okay. K minus 1. It is written degrees of freedom K minus 1. If it is between, say suppose X and Y, there are two variables. So it is the 2, it will be 2 minus 1. Okay, so suppose 1 2 3 4 5 1 2 3 4 5 in X there is 1 2 3 4 5, and in Y also there is 1 2 3 4 5. Then the total values are 10. Okay, so then what we will do, n minus k. So total is equal to n minus k is 8. Okay, like that we have to consider the degrees of freedom. With this calculation, with this formula, we can calculate the ANOVA. Okay, hope you have, and this is one of the formula, one of the calculations with some example that I have taken and to represent the decision made out of this. Okay, out of the values that I have calculated. Okay, not necessary now to discuss all that, because we have to move on to some other things like the various tools and techniques. Okay, these are some of the basic concepts of biostatistics. Coming to the fourth part that I have told you in the beginning. Okay, the relationship between the data. Okay, what more frequently, more commonly used is the correlation coefficient values. See, correlation, relation between the organisms, between the variables, between the parameters. Okay, correlation existing. So there is a relationship between the x and the y values. Okay, so in that case, how it is, how best it is suited, how best it is related, that we are going to calculate or to be discussing now. Okay, this is one thing called as coefficient. 1 minute, guys. 1 minute. Please give me a break.
Sir, next week my brain number.
Sir, congratulations, sir.
Thank you, sir. Bunny ಇಲ್ಲಿ ಆರಾಮಾಗಿ
ಸರಿ ಸರಿ ಸರಿ.
7:10.
ಯಾಕಂದ್ರೆ ನಾಳೆನೂ ಯಾವುದೋ ಒಂದು ಇತ್ತು.
ನೆಕ್ಸ್ಟ್ ಸಲ ನೆಕ್ಸ್ಟ್ ವೀಕ್ ಸರ್.
ರೈಟ್.
ಥ್ಯಾಂಕ್ ಯು ಸರ್.
ಥ್ಯಾಂಕ್ ಯು.
ಖಂಡಿತವಾಗ್ಲೂ.
Sure, sir. So I was talking about the coefficient value. So it's not the direct value. 1 2 3 4 5 1 2 3 4 5 there. So this is not those values are not there in the coefficient value. It is a value that is going to represent the relationship between the organisms, or between the variables, or between the parameters. Okay, so this correlation coefficient values are lying between -1 to +1. It will never go beyond 1, either in the minus case or in the plus case. You can see this simple line, -1, 0.5 and +1. So there may be 0 also. Okay, there may be 0 also. Say suppose this is 0, minus 0.5 and -1. Say suppose this is 0, this is 0.5 and +1. Okay, +1 is positively correlated, -1 is negatively correlated. Anything towards the negative, okay, anything towards the left region is the negative correlation. Say very, very simple thing. Okay, one is going up, another one is coming down. It's nothing but the negative correlation. There is in the both the cases. So one is obliging thing, other one is not obliging. There is nothing but negative correlation. So the highest value will be
-1 only. It will not, it will never go more than -1. Okay. Similarly, for positive correlation, one is going up, another one is also going up, or one is going down, another one is also going down. So in that case, it's nothing but the positive correlation. Okay, so the highest value, the coefficient value will be +1 only. If it doesn't obey any rule, if it doesn't obey, or if it doesn't have any kind of the respect to the variation in the first parameter, okay, then respect or influence of the with respect to the first or the second parameter, then it's nothing but the null correlation. There is no relationship. Okay, so it's like that. There are three types, or the three different ways that you can calculate these correlation coefficient. One is scatter plot. It's very, very simple. In your school days also, you have done this in a line chart or scatter plot. How the x values are going to, if x values are there, how the y values are going to be changed. Okay. In graphical representation also, you know, x is horizontally written and y is vertically written, right? So it's very, very simple, and one common thing that one have to follow to represent the graphical representation over here is x is always an independent value, y may be dependent. Okay, if you have both independent as well as dependent values, x must be, or your independent values must be represented on the x-axis, but on the y-axis you have to represent only the dependent values. Okay, in some cases it may be both the values are independent. In that case, more vulnerable you can put it on the y, and less vulnerable you can put it on the x-axis. In both the cases, both are independent. So here also, if both the cases are independent, if more independent, or if you feel that the influence of independent parameters, or if the values are moving towards the naturally available values, okay, so those values you can go and put it in the x-axis, the other one you can put it on the y-axis. This is a rule actually, then only your graphical representation is going to be fulfilled, or it is complete. Okay, so like that, the scatter plot. So the dots are rising from the diagonal, rising in a diagonally way, so it is positively correlated. Or if the dots are coming down from the y-axis towards the x-axis, then it is the negative correlation. If the dots are very random, it's neither moving any of the diagonal ways, then it is nothing but the null correlation. Okay, so it is like that we can define. Another thing is Karl Pearson's correlation coefficient. If none of the methods have been mentioned, then by default you have to go with the Karl Pearson's correlation coefficient only. Or it may be called as, sometimes it is also called as Pearson's correlation coefficient. Okay, so this Pearson's correlation coefficient is very simple and very commonly used for any kind of the data. Okay, so there are some set of the formula are there, and that formula you have to follow, and you can put your values in Excel and write your formula, and it will give the correlation values. Okay, so it will be the highest value must be 1 only, either +1 or -1. Another one is the Spearman rank correlation, where if you have the discrete values, okay, say suppose 1, 7, 18, 25, 30, 32, like that, if the discrete values are there, then what you have to do, you can assign the ranks to the values. Okay, say suppose 1 is the first rank, 2 is the second rank, like that you can assign the ranks either in increasing order or in the decreasing order. You have to assign the ranks to that. Okay, but you have to follow one pattern only. One case increasing value, another case decreasing, you cannot do. In both the cases, you have to follow one of the rule and make the calculations. And that is nothing but the Spearman rank correlation coefficient. And these values are also lying between -1 to +1 only. Okay, so in these graphs you can see first one, large positive correlation. If this is like this, then positive correlation. Medium positive because it is slightly down, and in the 0 it is up, and here the dots are spread out. Okay, spread out, not exactly lying on the line. Okay, the third one, small negative correlation. It is a negative correlation. It is coming down from y to z, sorry, x. Okay, but it is why it is called as a small negative, because the spread is too much. The values that are spread, the dots that you can see there, spread is too much. But of course, if you could see this fourth picture, so here the weak correlation. There's no correlation, because though the line we have put, but the values are lying here. They are moving away from the diagonal line, and either the case you can see the negative or the positive way, positive path or the negative path. Both the cases, the values are distributed in a very undisciplined way, and there is no correlation over there, or you can even call it as weak correlation. This is the formulas and all, you will get it very easily in any of the reference books. Okay, so not necessary to discuss about all these things, but we should know about how it is going to function and what is the interpretation after getting some of the results. Okay, that's very important. And nowadays, why do you need the formula? You don't need to remember the formula. Okay, no need to remember the formula. Not necessary to remember the formula, because there are some thousands of formulas out there. So why do you have to remember all that? But we should know about which formula to be used and what could be its interpretation or the decision that you are going to make out of that formula. That is very important than to remember or memorize the formulas. Okay, so not necessary about that. That's why some of these programs are there. That I will link these things to that programming things. Okay. So next one is Spearman that we have discussed. So this is the formula. Not necessary to remember or memorize this formula. Okay. So there are formulae, so you can make use of these formulae and get the decision or the interpretation out of it. Okay, so in case if you don't want to make any of the we don't need to remember the formula, what we have to do, we can write some of these programs. Okay, programs, what we have to do, we can write some of the programs of what our wish, our aim, our related formula, our related objective. Okay, for that you have to write the formula and get the interpretation or the decision out of it. Okay, so that's the programming thing. That is also very simple. It's not very ತುಂಬಾ ಕಷ್ಟವಾದ ಕೆಲಸ ಏನಲ್ಲ, right? It's also easy, but only thing is you should know what to write and when to write, for which one and which not. Okay, like that it is there, and we will discuss the SAS programming things after these two slides. Okay. One more related kind of the estimation or the calculation is the regression analysis. So here comes two things. One is regression coefficient. Okay, that is the constant that we have to calculate. It is also represented with the letter B. Okay, probably you might have heard or seen or used one of the formula, that is y is equal to mx plus c. That is the slope equation. Everybody knows, every engineer knows about it. Okay, the slope equation. This is also exactly the same thing. Okay, its interpretation is different, and that too in biological interpretation it's very, very different. Okay, the regression coefficient is the constant b in the regression equation. See the formula, y is equal to a plus bx, or bx plus a, that you can say. That tells about the change in the value of dependent variable. Dependent variable is x, right? So I told you in the graphical representation also, dependent variable is x, corresponding to the unit change in the independent variable. So if x is going to change, how does the y is going to change? What is the pattern, or what is the level or the degree of change in the y that can happen with respect to the change in the x? Okay, that is nothing but the regression analysis. And how to calculate the b and a. Okay, well, first you have to calculate the B, and with the help of B, you have to calculate A, and apply the formula, and after that you can you will get X values, and substitute the X values in that formula, and you will get the Y values. Put it in a graph and see how they are related. Okay, that is the regression equation or regression coefficient values to be calculated. Guys, do you have any doubts so far? With this preamble, we will move to the SAS. Okay. The statistical analysis system. Probably you might have heard about this SAS program. Okay. So SAS is nothing but statistics analysis system, statistical analysis system. Okay, so there are various tools. Probably the best tool in the world is the MS Excel, I can say. Okay, it can link from one cell to another cell, one row to another row, one column to another column, one sheet to another sheet, one file to another file also. Okay, so if you keep the values in one row or one column, okay, another column is also filled with, by having these two variables or the two columns, the data set. So what you can do, you can write any kind of the formula, you can write any kind of the algorithm. Okay, you can write in a more advanced way, you can write any kind of the program to analyze those two variables. Okay, Excel is such a wonderful tool. MS Excel I'm talking about. MS Excel is such a wonderful tool. You can get any kind of the calculations. Okay, so the modification of that, say suppose you need to write a formula where which cell you are going to consider, which are the cells that can be omitted, which are the cells that can be defined properly, which are the cells that can have more influence or less influence. So these kind of the factors will be there. For that, the modification of that are development of some programming languages towards the statistics have been taken place. Okay, there are various tools also. Okay, like your Excel, there are various tools. Say suppose one is SPSS. Okay, so you have two windows in a file. There are two windows. One is the variable window, another one is the output window. In variable window itself, you have two different sheets. One is data view as well as another one is variable view. Okay, so like that, the tables will be there, and in the tables you have to enter the data. Those data are nothing but, each column is nothing but one variable, or you can define it as a particular parameter, and those parameters you have to calculate. You can subject these parameters or the variables to your calculation. Okay, so in SPSS, what happens? In SPSS, there are, say suppose there are two variables. You are going to click on the correlate. You will select which type of the correlation that you want. You will select which are the variables that have to be considered for the correlation. It's all just on a click. You can click and get the results or the output out of it. Okay, finally, interpretation of that result is in your hand only. Okay, so more advanced. Suppose I don't have much time to put the data in a form of a table, or I don't have much because of the huge data or the big data, or maybe the large amount of the development of this data. So we don't have the patience, or we don't have the skill set to put the data in its required way. What you have to do then? You have to depend on some of the programming languages. Okay, you all know about what is the programming. So probably you might have heard about the programming languages. Okay, probably you know about the algorithms. Okay, one more thing is algorithm. Programming language is a formal language, okay, that will comprising a set of instructions that produce various kinds of the output, okay, in a programming language. So you are going to give some of the instructions. That's nothing but the algorithms, right? Instructions. You are going to say this is a pattern that I want. You are going to say this is the pattern that you have to work with, or you can say this is how the you have to relate the values, okay, or this is how, which is the parameter that is highly influential, okay, like that you are going to tell. That's the algorithm in a program, right? So the programming language, it consists of many kind of these algorithms previously defined, which will be present in the library, okay, and these will be are the instructions, are the set of instructions, okay, that will give the different types of the output. Why different types of the output? Because you are going to define that which kind of the output you need it. Either it may be a correlation, or it may be arranging the values in an ascending order, or arranging the values in a descending order, or arranging the values with respect to some of its patterns, definite patterns, like that you are going to put it up. Okay, for that SAS and R programming. Okay, SAS is a different programming language, and R is a different programming language. Okay, R is freely available, but SAS is not. Okay, guys. So, but in our syllabus, we have to discuss about SAS because it is there. It is prescribed over there. Let us see how best the SAS language is going to help for the calculation purpose. Either for the calculation purpose, like whatever we have discussed so far. So all this can be done using the SAS. It's very simple language. Okay. So you probably, if you have experienced about any of the programming languages, so it will be very easy to execute any simple set of information to get our interpretation out of it. Okay, so let us see how the SAS data sets are there. We have to understand how the data set is all about. Then we can go with the calculation. Then you can write correlate between A and B, okay, and run semicolon. Then it will run and it will give the values of A and B correlation. Okay, so what is the correlation between the A and B values are there? So it's going to give. But before that, we have to understand what is the data set in SAS. Okay, and how does it's going to look, and how what are the simple kind of the programs that can be executable. Okay, for any kind of the programs or the algorithms, no need to worry to build a new story. Okay, so there are various algorithms that are available. You can take that, and if you want any changes to be made, you can change it, and you can execute your values. Okay, so the SAS data sets, examining the SAS data sets. The data sets is something like it has two sections here. One is descriptor sector and section, and data section. Okay, the descriptor section is something like this. You are going to describe the data. Okay, so observations, how many observations are there, variables, how many are there, okay, member type and engine created, when it was created. So like that, the data information will tell you about when it has been done, how it has been done, how many number are there, whether these are correct or not. So like that, the descriptions can be made. So deleted observations, added one, okay, so like that, compressed one, sorted. So like that, the description section is going to tell you about the data. Okay, so that's the descriptor section. But majorly we need the data section. We'll work in the data section in a majority way. Okay, but we cannot omit the descriptor section also, because once you have defined the correct descriptions in the descriptor section, then only you can say you have defined. If you define the correctly, then only you can give the correct interpretation or the correct decision on your analysis. Okay. Analysis can be done by using any of the tools. Okay, like I told you, SPL, SPSS, and Excel. There are various other tools also. Okay, other tools may be GraphPad, StatCraft, Systat, okay, Krebs, uh, uh, you know, MATLAB, uh, Minitab, okay, like that. There are some hundreds of tools that are available. Some of them are freely available also. Okay, you can use these tools also and put the data and get the interpretation out of it. But in SAS or in R programming, what you have to do, you are going to define that. You are going to tell which kind of the data is it and how you are going to get the interpretation out of it. If you define it properly here and make the analysis are also in SAS as well as in R, it will be in our hands. Okay, so whatever the calculations that are going to happen, it will be based on our will and wish. Okay, not say suppose in SPSS or some other tools, whatever the calculation that will happen, the calculation part, the algorithm part, that we don't know whether it has considered all the values or not, that we don't know whether there might be some kind of the iterations that we don't know. Okay, but here in a descriptive section or in the data set, you can understand whether the correct calculations have been done or not. Okay, the descriptor section that you can see now. Okay, various types of the information about that data have been put in this. Okay, that's the descriptor section. Next comes the data section. So the data section will tell you about, see, you can see the faculty ID. Okay, whatever the sections are, the different types of the sections that are experience or the expertise that they have. Okay, and satisfaction with the department. How many number of anthropology expertise? 5 people. Family and counseling studies 4. So like that, the information is properly managed or properly dealt in the data section. Okay, you can even add many columns also. Okay, you can even add many columns for this and get the data section properly for the further analysis. So attributes of variables may be like name and type. Okay, in this example, okay, students are in a college database. Okay, faculty expertise, faculty relationship, or satisfaction level, or how the teaching levels are, what is the percentage of the results, like that, if you could talk about. So the various attributes that of that variable can be mentioned. So it may be the name or the names. Say suppose in the previous slide, so we saw this nursing, English, economics, history, finance, like that, right? So like that, you can mention the name or the status or the expertise and so on. Okay, next comes the type. It may be numeric or character. Okay, numbers are the character. Status of this example may be T, TT, PT, or any number that you can say XYZ or ABCD, A plus, A, S grade, A grade, B grade, like that you can give. And satisfaction may be numeric. 1 to 5 is highest. Okay, 1 or 0 is the lowest. Like that, you can give the rating levels. Okay, or the satisfaction that can be represented with the numerics. So there are some of the variable features or the attributes of those variables. However, you have to go, or however, it's not like whatever the way that you wanted to give. So when you are talking about the satisfaction, you have to give the numbers, numericals only. Okay, status, either it may be a number or it may be a character also. Okay, so SAS data set terminologies are like this: variables, observation, numerical data, and character data. Variables may be columns in the SAS data set. Variables, okay, variables are the parameters that we can say. Okay, they are the columns. Observations are the rows in a SAS data set. Observations, whatever you are going to do, they are the observations. So like, I'll go back to this slide. Okay, the column serial number 1, 2, 3, 4, 5 up to 30. Okay, and department number, satisfaction levels. Okay, so these are the columns that you can see. They are the variables. Okay, but the observations are in this row. So it means nursing. There are 5, 27, T, 0, and date. Just see this date, don't see the date now. So if you, they are the observations. Okay. So say suppose English, 5 is the satisfaction level, 27 number are there, and T is the another satisfaction level, D is the degree. Like that, you are going to tell. They are the observations or observation levels. Okay. They are the rows. Numeric data, values that are treated as numeric and may include 8 bytes of floating storage for 16 to 17 significant digits. Any numbered data. Okay, so they will be coming under the numerical data, not necessarily 8 bytes and such stuff, because now though, because of advancement is something like every 6 months, every 2 months. Okay, every 6 months you are going to get the updates. Okay, not necessarily only the 8 bytes. Okay, so it can increase more also, 16 to 17 digits se bekagilla ಈಗ. Okay, so this is a pay version. SAS is not free. It's like R. SAS is a paid version. Okay, if you want to learn SAS, beta versions may be available, but beta versions will not give you the sufficient knowledge on how to learn it. Okay, SAS is like that. And if you want to learn R is one of the best programming language, even more advanced than the SAS, and that is free now. Okay, next comes the character data. It is a non-numeric data values, such as maybe the letters, numbers, special characters you can consider, or the blanks. Okay, that you can consider, and they are nothing but the character data. It may be stored, okay, with various varied length, or 1 byte is equal to 1 character that you can also make. These are the preamble towards the SAS. Okay, SAS dataset and variable name criteria is maybe like this. If you take the SPSS into consideration, it will take up to 32 characters. Here also it's taking up to 32 characters. Okay, but in R it can go beyond that. Okay, so it can be uppercase, lowercase, or the mixture of the cases that can happen. Okay, to represent the variables, and they are not case sensitives. Okay, it cannot start with the number and cannot contain special characters or the blanks. Okay, that's in SAS, but in R you can say you can even have the characters as well as the blank spaces. Okay, it can start with a letter or underscore. You can, but if you, wherever it's very common sense or common to understand the thing, that wherever underscores are mentioned or a mandate kind of thing, there you should think that there should not be any space. There should not be any blank space. Either you have to go with the underscore, or you can start with the underscore, or you can, in between the words, you can give the underscore, but you cannot give any blank. Okay, say suppose user ID, user underscore ID you have to give. It's not user space and ID. Even the comma you cannot, it cannot, and user hyphen and ID, it's not going to take that also. You have to give user underscore ID only. Okay, or you can give user ID continuously, there is no space at all, USER ID. Okay, like that you can give. Mostly all the date formulas are, date formats are in the numerical only. Okay, so they are all numericals only. It's not like January 1st, JANUARY or JN. It is not going to consider. Okay, and there are a number of days. Okay, as a 0 point. Dates. Why we have to give the dates? If in some cases we can give the dates, or in your experiment, if you are comparing the values from different seasons or different years or different periods or something of that sort, if it is in the time scale, the temporal scale, then only you can you have to give the time. So date and time. Okay, so when you're talking about the date, it will automatically consider those dates and number of incidences or number of the parameters that have been changed, and the variations in those parameters can be easily calculatable or computable. That's why we have to give the dates. But those dates are in the must be in the form of numeric data. For example, you can see here in the last column, you can see 0 to 12 and 2007. It means that it's day, sorry, DD and YYYY. Okay, that's how the representation has been done over here. First is the month and the day and the year has been represented. Okay, so that it is like that, and time is scored as a number of seconds since midnight. So when you are talking about the time, either 24 hours or the 12 hours range you can give, and even you can even mention the seconds as well. Okay, the significance of mentioning the time and date over here is to compare the values, as we discussed earlier, no, the correlation coefficient or the regression analysis or maybe any other time series values. So for that, if you want to compare those values or get the interpretation out of that, you have to mention the proper time as well as the date. The time should also contain the hours, minutes, seconds to be considered, right? Missing data in SAS values. So generally, the missing data is one of the biggest issue or biggest kind of the point to be discussed over here. Missing values are valid values here. Okay, missing values are something like there may be certain errors in the data collection. There may be certain errors in the process of processing of the data, or maybe errors in the human or any of the system errors. Okay, so these errors to be nullified, you may have to add some of the values that will be in the name of missing data or missing values. Okay, so most of the cases when we are intended to do the or calculate the missing value or missing data, they are the valid values only. If not, if it is only to understand the pattern or maybe the maybe just to exploration of, not exploration. One second, sir. ಇದು ಒಂದು ಜೀವಿಕೆ ಸರ್ ಒಂದು ಇದು ಮಾಡ್ತೀವಲ್ಲ, ಗ್ರಾಫ್ ಹಾಕ್ಬಿಟ್ಟು ಪ್ರಿಡಿಕ್ಷನ್ ಮಾಡ್ತೀವಲ್ಲ, ಅದು ಏನೋ ಅಂತೀವಲ್ಲ, ಅದೇನದು ಅದು ಅದಲ್ಲ ಸರಿ. ನೆಕ್ಸ್ಟ್ ಇಂಟರ್ಪ್ರಿಟ್ ಮಾಡೋದಕ್ಕೆ ಏನೋ ಒಂದು ಟರ್ಮ್ ಇದೆ ಅಲ್ಲ ಸರ್, ನೆನಪಾಗ್ತಿಲ್ಲ. ಕರೆಕ್ಟ್. ಒನ್ ಮೋರ್ ಟರ್ಮ್ ಇಸ್ ದೇರ್, ವೈ ಆಕ್ಸಿಸ್ ಅಂಡ್ ಆಬ್ಸಿಸ್ ಅಂಡ್ ಆರ್ಡಿನೇಟ್. ಹಂಗಲ್ಲ, ಈಗ ಪ್ಲಾಟ್, ಈಗ ಒಂದು ಪ್ಲಾಟ್ ಮಾಡ್ತೀವಿ ಸರ್, ಎಕ್ಸ್ ಜಾಸ್ತಿ ಆದ್ರೆ ವೈನು ಜಾಸ್ತಿ ಆಗಿ ಲೀನಿಯರ್ ಇದು ಬಂದ್ರೆ ಲೀನಿಯರ್. ಇದನ್ನ ಏನಂತ ಕರಿತೀವಿ? The graph will varies linearly with respect to y and x axis. There is one more term in the start. I'm not getting that word, friends. Maybe at the later stage I will tell. When y varies, x also will varies linearly with respect to y. Correct. But for that there is one term. It starts with E. I'm not getting that now. Okay, sir. I'll ask. Thank you, sir. Okay. So for that, if you have missing values can be considered. Okay. But these missing values can be understood, or missing values can be obtained by iterating the data. Probably know about the iteration of the data. So it may be like, say, suppose 1, 2, 3, 4, 5, 1, 2, 3, 5, 1, 2, 3, 1, 2, 4, 5, like that. The data is there. The missing value in first case is 4, and the second case is it is 3. Okay, how does it is going to be calculated? By calculating all the means, or maybe the differences among the variables. Okay, so with that we can calculate the missing values, or we can even iterate by using the algorithms over there. Okay, for character data, the missing values are displayed as blanks. Okay, for numerical data, it is display the missing values are displayed as periods, or the year, or the schedule, or the time series. Okay. How the syntax is there? It's very simple. Probably you will be knowing about the C programming. You will be knowing about many other programming languages also. But here in SAS syntax, it is very, very simple. If you download the SAS, if beta version is available, if you want to purchase the SAS, you probably may have to pay certain dollars and get it. One is, once you install that, you will get the libraries. The libraries will tell you about what is the syntax, how does the syntax is going to work, what are the different kind of the algorithms that you can put, or the programs that you have to write for some of the common kind of the variables, some of the common kind of the analytics. Okay, maybe for the correlation, or for the slope equation, or for estimation of some of the graphical things, and so on. Okay, the syntax, the simple syntax over here are like this. The statements in SAS are like the sentences. Okay, it's not like print a word, hello world, and colon, semicolon, and close the bracket, open the bracket. If you miss one of the one of these syntax, and your entire program is not going to execute. Not like that. Okay, that's why it's very simple. Simpler to that is the R programming, people. R programming is the free one, and it is much simpler than SAS, I can tell you. Okay, so but still, now today we will discuss on this SAS. They are like sentences. So if what you want to do, say correlation coefficient. I want correlation coefficient. Don't say I want, but correlation coefficient colon, like that you have to give. The punctuation though is a semicolon. Okay, rather than a period, it is dot. Okay, most statements, but not all, start with an identifying keyword like a proc, data, label, options, format. These are the some of the keywords. They are the identifying keywords. Okay, so it's going to identify which statement I have to consider. Okay, proc is for the process, data, label, options, formats, like that. And these are also there in the library of that SAS. In SAS libraries, you will get these also. The statements are strung together into sections similar to the paragraphs. Okay, the statements are strung together in one string. You can, in one stretch, you just put the comma, not comma, semicolon, and put your statements. Okay, say I will go back to this table. Say suppose 1 semicolon anthropology is equal to 5. So here what happened in anthropology, where it will come? It will come in the department. Faculty ID is equal to 1, semicolon, department is equal to anthropology is equal to 5, semicolon. Like that you are going to write. Okay, it is like a paragraph. In these are some of the paragraphs in a Windows operating system. Okay. And that will be end with the word run and a semicolon at the end. After all your statements, below that type the word run and semicolon, and then it is going to execute. Okay, very simple kind of the statement over here that you can see here in this slide. Okay, it's very, very simple. Process data. Okay, this is the common one. This is by default in the syntax window. You will get this. Okay, so label ID is equal to faculty ID. Department is equal to department. Satisfaction with the department. Years, that is equal to years employed at the university. Status is equal to faculty status. Degree is equal to degree. Date is equal to date. Okay, please read this again. Okay, just have a look at this, and we'll go back to the previous table. This is a table. For this, for the production of this table, or the making of this table, this is the syntax that we have to use. Guys, got it? It's very simple kind of the program that you can write. It's not very great, right? Just department is equal to department. Satisfaction is equal to satisfaction with the department. How with the years. Department satisfaction over here with the years, 5 years, like that. The syntax can be written. Okay. Options is equal to 100 no date. Run semicolon and enter. That your program is executed. Okay, this is the simple kind of the syntax available for SAS. Okay, if you want to run the correlation between the variables, define the variable and give the correlation. It's as simple like that. Okay, coming to syntax rules, how the syntax rules are there. 1 minute, guys. I'll just drink water and come back. Hello. Morning. Hello. Big. Tell me. ಇವತ್ತು ಮಾಡ್ತೀನಿಮ್ಮ, ಇವತ್ತು ಮಾಡ್ತೀನಿ. ಇಲ್ಲ ಮಾಡ್ತೀನಿ. ಏನು ಸ್ವಲ್ಪ ಬಿಸಿ ಇದೆ, ಮಾಡ್ತೀನಿ. ಓಕೆ, ಈಗ ಕ್ಲಾಸಲ್ಲಿ ಇದ್ದೀನಿ, ಆಮೇಲೆ ಮಾಡ್ತೀನಿ. ಇಲ್ಲ ಮಾಡ್ತೀನಿ, ಇವತ್ತು ಮಾಡ್ತೀನಿ. ಈ ಕ್ಲಾಸಲ್ಲಿ ಇದ್ದೀನಿ, ಆಮೇಲೆ ಮಾಡ್ತೀನಿ. The SAS syntax rules are something like this. They are also very simple and easy. Okay, the SAS statements are format free. No need to follow a definite format, and one simple comma or semicolon are going to disturb you. Not like that. Okay, one or more blanks or special characters are used to separate the words. Say suppose if in Excel, for example, if it has to identify certain date formats or the numbers. Okay, say suppose you are writing some 12 numbers in a continuous way, or 15 numbers in one string. What it will take? It will take to the exponential values. Okay, but here not like that. Just give one space, it will understand. In Excel, suppose if you have in the beginning, if you give some space and start the string, it will, when you will put the array, so it will go to the at the end of the array or in the beginning of the array. But it is because there is a space over there and it has got impact. But here, if you could do those kind of the mistakes also, it's not going to consider all that. It will nullify your mistakes and it will consider the word directly. Okay, so in one paragraph you are going to give the syntax, or maybe 2 or 3 paragraphs you can give the syntax, and finally run semicolon and enter. You will get your work done. Okay, one or more blanks or special characters are used to separate the words. They can begin and end in any column. Okay, to define that. So if you want to define the table in a proper way, what you can do, one below the other, you are going to give the syntax. Okay, a single statement can span multiple lines, right? So one single statement can give you the multiple lines kind of, right? The several statements can be on the same line. See this one. So this satisfaction is equal to satisfaction with the department years. Okay, so here in this one he has made all different statements in a different line. But say suppose degree is equal to degree, comma, and write date, push it over here, no worries on that. So it will consider that, right? So several statements can be on the same line that you can have. Say suppose this one is here, all the lines are here. See, you have a glance of this slide, and we will come to the next one. See here, it is label ID is equal to faculty ID. Okay, one space, department is equal to department, satisfaction is equal to satisfaction with the department, years is equal to years employed at the university. There is just a space, status is equal to faculty status, degree is equal to degree, date is equal to date. Okay, option is 100, no date, and run and enter. You will get your program is going to run. Is it a tough date? Not at all. Okay, it's not at all a tough job. It's very simple and easy. Using the free, what you have to do, you have to practice. Minimal practice must be there to understand this, because where it has been created, how it has been created, that also have to be understood, right? We'll come to that. Using the free format syntax, that rules of essays, though can make it difficult for others to read your program. This can akin to writing a page of text with little attention to the line breaks. Most of the cases, what happens, if there is a rule to give the break to the program or the lines, then it's very difficult. Most of the cases, what happens, people are going to write the programs in a continuous way. Only thing, just like our words that will be typed in the Microsoft Word. Okay, so like similar to that, it's very easy. If you give the concentration towards the splitting of the words, towards the splitting of the lines and all, or making the paragraphs and all, so it's a tedious process, and there is a drag, or there is a delay in the process. So to avoid that, so the best thing is to run the program in a paragraph manner. Okay, this is the formatted one that we have seen in the earlier also. Free format syntax rules. Okay, so this is already we have seen. This is the correct formatted one. This is a paragraph one. Okay, both the cases that you can write. Some of the comments. Okay, so to begin the comment, so this is a slash and star. Type your comment text and type slash and star and slash to end the comment. End, begin and end. It's very simple, no? Not very great thing to be remembered. Even in R also, to begin, slash and slash is to end the comment. Okay, or type an star at the beginning of the line. Everything between the star and the semicolon will be commented. Ist kotre saku, so it will be commented. For example, in file tutor dot date and semicolon. So it's nothing but it's going to consider the in file, whatever the either in the Excel or any other formats, and tutor dot date is going to, this is the format, and it's going to analyze that. So this is going to end, semicolon is going to end. Alternately, highlight the text that you would like to comment and use the keys control to comment the line. So it's just like a shortcut way to do that. And to uncomment the line, highlight and use the control shift keys. Okay, control if you use, then and highlight, then it will going to comment. If you use the control shift and then the highlight, is going to uncomment. It's going to end over there. It's like a shortcut usage in a NSS comment line. Okay, so options ಬೇಕಾಗಿರುತ್ತೆ ನಮಗೆ. Okay, so what you have to do, this is one of the syntax that you have written. Options, just like this, you are going to highlight. Okay, and run, then it's going to run and give the program. How does it is going to look? If you open the SAS program, then 3 windows will come. Okay, this is the Explorer. Then even if you can minimize this, okay, Explorer, that will have many of the files. Okay, and editor, this is the editor one, and this is the log is about the when you have logged in and how does it's going to be work, and what are the programs that you can work, and all will be here. But where you are going to work, you are going to work in the editor. Okay, so enhanced editor window. This is the editor window that has been enhanced over here. So what are the outputs once you run it? What are the outputs that you are going to get? Here you are going to write, and you are going to get the outputs over here. Okay, your program script appears in this window. You can either bring it in from a file or type the program right in the window. In this window, in the below window, you can bring the file or you can type the program. Okay, so once the program is in this window, okay, you can click submit or the running guide. Okay, run at the type madi, enter madi, or you can give the submit button. You can put the submit button and it's going to be executed. Okay, the SAS log that will give the blow by blow account for the of the execution of the program. It includes how many observations were read and output. So the log will tell you about how many have been read and observed. Okay. For the consideration of the output, and also it will also tell you about the errors and the notes. If any notes are also there, it will to if any errors are there, that is also to be mentioned over here. Note the errors in red. Errors generally will be coming in the red. The output, once you are going to write the program or add the program here in this window, so and submit or run, then your table, the formation of the table or the output window will be displayed over here. Okay, in SPSS also, once you are writing the once you are executing the program or the calculation or the computation in your variable window, or even in the descriptive window also, you can do that. So what you will do, you will go to the analysis and type and select whichever the type of the analysis that had to be carried out. Then what you will do, you will execute it. You just say submit or calculate, then it's going to give the output window. So here the output window is going to put all the values in the form of this table. Okay, that's what has been asked, and it is going to give that. Okay, so for any kind of the help or the requirements, what you can do, you can have the SAS library. 1 second, guys.
I wanted to ask if I could work on this to the technical.
I'll close this. You'll be here, no? 5 to 10 minutes.
Okay,
Not more than.
Thank you.
Okay, the data libraries are there. That's, I think, in the previous window also you have seen this. Say in the Explorer, where you can see the in the Explorer where you can see the data libraries. So here what happens? So you can see the libraries, file shortcuts, and favorite folders. So if you explore this, so you'll get the minimal, the most commonly used kind of the programs. Okay, just copy those and run it. For example, if you want to calculate the correlation coefficient between some of these parameters, what do you have to do? Write a program, correlation among the variables, correlation among which are the variables that you have to type, and then run, or you can submit. Then it will give run it and will give the values of correlation coefficient values in the output window. The SAS data sets are files within those drawers. Okay, libraries, if you go, or data sets. Note that icons on the SAS library match the metaphor. In order to assign the drawer, you assign a library reference name libref. Okay, suppose if you no need to copy or no need to, if there are too many things are there, what you have to do, library ref that you can give, and it's going to take the files from that library reference. Okay, there are 2 drawers already in your library: work temporary and saucer permanent. Okay, work is for the, you can add the data, and in the saucer it's already permanently or with by default those values have been mentioned. Okay, so you can also create your own kind of the libraries or the drawers using the lib name statements. Okay, establishing the lib name, it's very simple. If you want to create your own kind of the programs, if you are using the similar type of the programs for longer duration or the for more and more computational techniques, then what you can do, you can establish your own kind of the library name, and you can add it to the library set in the editor window. It will come, and you can use it for the further process also. Similarly, if this SAS have got these many options, the R programs still have got more number of the options. Okay, so it is like say suppose you have a set of the information about the climate change. Okay, so the data is there, and drastically it's going to change the climatic factors or something of that sort. You are going to provide the link to some of the GPS stations or something of that sort with respect to with the permission only, with respect to the temperatures or the rainfall and so on. Okay, so it is linked. Once you will change the date, okay, say suppose next month 25th, what could be the rainfall? Okay, just change the date, enter, and you are going to sorry, you are going to get its interpretation. Okay, so like that, maybe it's going to be rain or very shiny or sunny days or something of that. So those kind of the information that you are going to get once you just change the date over there. Okay, that's the advantage in the R. Okay, but only here you can just create the libraries and keep it for your future references. Okay, view table window or the output window will be like this. Okay, so it has the serial number, ID, faculty ID, different disciplines or the department satisfaction levels, and so on, so forth. Okay. In the one of the previous slides, I have told you about proc, data, formats, and some different types, right? One such is the proc. Okay, so if you want to say the process, various types of the process and all, there it says analysis of variance table is mentioned over here. In this, what has been model in the what's the degrees of freedom? You just describe that. Okay, so you just give the sum of the squares, mean squares. Okay, it will give the f value. Okay, no need to calculate the f value and even the probability value, the p value. P value is also been mentioned over here. It is less than 0.001. Okay, so we know about the degrees of freedom. What are the what is the degrees of freedom and all? We know we can even calculate the sum of squares earlier. Before that, before going to the analysis ANOVA, what you do? You can calculate the sum of the squares and mean squares and put those values R. No need to put that values. You can just link that value here in your program and run and put run, and it will give the f value. Okay, and automatically it will give, based on the degrees of freedom, it will give the p value. It's not called as PR, it's mentioned as PR, but in majority of the cases it will be mentioned as p. Okay, p is greater than f, that is the table value. Okay, from the this f value is greater than the table value. Okay, so like that, this simple the ANOVA can be done very simpler way using the SAS. Hope I have covered most of your that required topics. Guys, if you have any questions, please let us discuss. SAS is not going to end with this. It has many other features also. If you could get the licensed version, probably we can see even we can even work, and how I can show you how the changes are. If I write a simple program, how the changes can also happen. If I have to install that, I have to purchase that only. Okay, so and actually people are using R more effectively, efficiently than SAS, and I don't have that software in my system, and I can only tell you how it's going to work with some of the experiences. Okay, guys, and I can share the study material to either Manjunath sir or madam, Valarmati madam, and you can collect from them, and you can study for your exams. And that's done from my side.
Yes, for your very elaborate and wonderful. Students, if you have any queries, you can either post it in the chat box or unmute yourself and talk to sir.
Thank you, madam.
Our students, you can post the question to me later. I will discuss with sir and we'll get back to you. Shall we end the session, sir?
Okay, madam. Thank you, madam. Thanks for the opportunity.
So, on behalf of the Department of Biotechnology, the Oxford College of Engineering staff and students. Sir, I deem it a great pleasure in extending you a very heartfelt gratitude for giving an wonderful lecture on this event. Thank you so much, sir, on behalf of the Department of Biotechnology staff and students. Thank you once again, sir.
Thank you, madam. Thanks a lot.
Thank you, sir. Sir, you may log off, sir.
Okay, ma'am. Right.
Students, please fill the feedback. It is posted in the chat box. Please fill it.